DNA strand in the nucleus, and for replication during the S phase, the helicase comes in right here, and it creates a replicating fork. The single-stranded binding proteins also come attached to the backbone of the existing DNA templates in order to make strands not retwist again for replication to happen. So the helicase is undoing the hydrogen bonds, and the single-stranded binding proteins are coming with it. Then the primase comes, and that starts with the RNA and primer comes. So now there's a 3 end for the 5 ends comes. So now there's a 3 end for more to attach. And so there's nucleosides, and the A nucleoside comes in, and then the phosphates are chopped off for energy. And then the backbone is created, and the bond and the carbon is created. And then the DNA polymerase 3 keeps moving on the leading strand. And again, the phosphate group is off for energy to fuel the bond. And we're going to keep going. And the helicase keeps going and making the fork. And the proteins keep going. Okay, so meanwhile, while that's going, the lagging strand then, right here, starts where the primase comes and brings in an RNA primer. And so it brings in the primer, and so to go from the 3 to 5 direction, a 5 comes in and binds, and then again the phosphate group gets cut off, and then this keeps going. Primase again comes and brings in another RNA primer, and then the process again keeps going. Alright, and then here and here. And then these two then, this one and this one are Ozaki fragments as the lagging strands. And it needs the help polymerase 1. And so DNA polymerase 1 comes in and removes the RNA primer and replaces it with a nucleoside. And this comes in, and the phosphate comes off again. But then here, this binds and is with the backbone. But then ligase has to come in and bind these two together because they were not previously bound. So ligase comes in and does that. And then it keeps going in the same fashion. And we keep going until the whole thing is unwound. And these are up here. And so we're just going to keep going in the same fashion. So we are totally done. And this DNA polymerase 3 is going. And we're cutting off the phosphates again for energy. We need one. And then we go back to the beginning of the leading strands. We have to remove the RNA primer because that is not actually part of the nucleosides. So the DNA polymer 1 does that, but because only a 5 carbon left here, you cannot attach to a 5 carbon, and then this is the end. We end up with 2 different new strands of half old, half new, semi-conservative DNA.
The end.